So just to come back to a sort of slightly different kind of timeline, we saw something of the history of the lake just before, and that gives you a really good feel for how this lake might have been impacted over time by all kinds of different stresses imposed by people and their activities. As Lindsay said, the monitoring of the lake started a really long time ago and was initiated by the Freshwater Biological Association. There was some measurement and sampling in the 30s, but the monitoring really started in anger in the 1940s. And that's going out into a boat, sort of manual monitoring. I'll say a little bit more about that later. That's been ongoing ever since the 40s. In the late 1980s, that was the handover between the Freshwater Biological Association undertaking that monitoring and our predecessor organisations taking it over. Now, what's actually happened as well over time is that more and more approaches and methods to gathering evidence from the natural world have developed over time as technologies have developed. So when it came to the 1990s, we started to be able to supplement what we were already doing by installing sensors on automatic water quality monitoring buoys on the lake. So we could get really fine resolution data over day-to-day scales and hour-to-hour scales that open up the possibility of looking at all different kinds of behaviours in the lake, all different kinds of stressors. A little bit later on, we were then able to start looking at satellite data, and that really started in the early 2000s, looking at basically properties, optical properties of the water that you can see from space, and then trying to make inferences about the state of the lake based upon those properties. Going a little further on in time, a bit more recent, we've then been able to monopolise on developments in molecular techniques for monitoring. So perhaps many of you have heard of eDNA. That kind of work has been done here as well. If you haven't heard of it, it's essentially taking a water sample from an aquatic environment, extracting the DNA from all the organisms and the organic matter in that sample, and sequencing it to identify which species were there, essentially in the lake. And it can be an incredibly sensitive method. And that's been done actually to assess the fish populations in Windermere, as you can see, back in 2015, and was really insightful actually. Bringing us right up to date, even more recently, that's when actually many of you probably got involved with citizen science activities. There's been a real ramp up in those kinds of activities in community engagement with monitoring over these last few years. Firstly with the Blooming Algae app, which you'll hear a bit more about later, which you can have on your smartphone, whether it be Android or Apple, and you can take pictures of algal scums. And then more latterly with the big Windermere survey, which you're going to hear quite a lot about from Ben a little bit later on this evening. So not only has there been a really long story in terms of the monitoring of the lake and the gathering of the data, there's been something of an evolution in the way that we do it as well as new techniques have come online. And to just really restate what Lindsay said, this really is one of the most well-studied lakes in the world and is globally recognised as such by the research community globally as well. So it's quite an incredible feat, and we're very lucky to have all this knowledge, evidence and information to inform us on the state of the lake. So just quickly to remind you then, the monitoring that's undertaken just now is from 4 basins in the Windermere catchment. So Esthwaite and Blellantarn, we monitor those over that same timescale that I just showed you before, but also those 2 basins of Windermere. And we just touched on why that's the case, because they have quite distinct properties from each other. So we've always monitored those separately from each other so we can actually distinguish those differences in behaviour. And what's really nice about this is that in a really small geographic region, you've got lakes that are really quite different in terms of their chemistry and their properties ecologically, and really different in size and depth as well. So you've got a selection of lakes, and if you monitor them all in the same way, you can start to arrive at some quite interesting general understanding that might apply to other kinds of lakes too across the world. So what does this monitoring look like then? Well, right from the off, way back in the 40s, it was boat-based surveys. So people are going out in a boat, they're collecting water samples, bringing those back to the laboratory and analysing concentrations of nutrients, concentrations of indicators of the amount of algae growing in the water. They're collecting samples of zooplankton, that's the little invertebrates that live in the water feeding on the algae. But also taking sensors and probes out there to measure things like temperature, oxygen, pH, and lots of other different parameters that help us to understand the lake. So quite a cross-section of physical, chemical, and biological properties are being measured every time we go out. That sampling is every 1 to 2 weeks across that whole time period, year-round, pretty much whatever the weather, so it's either glorious or really a bit testing, depending on what the weather's doing at the time. But what that does is it means that we can look at really long-term changes in the state of the lake, but because we're going year-round, we can look at seasonal changes as well, and we can look at how the lake's changing from one fortnight to the next to just get a bit more of that shorter-term variation in the properties of the lake. And then with the advent of the buoys a bit later in the record, even finer scales still. So we're generating knowledge and evidence that allows us to look at things happening over lots of different scales. One of the really interesting features of this monitoring scheme is it wasn't necessarily set up to be a long-term monitoring scheme. So actually we owe an awful lot to some really well-respected and established scientists, John Lund and David Leckrem, who really kick-started this initiative because they wanted to know about the factors that drive the seasonal growth of algae in the lake. Maybe not the same ones that people are concerned about just now, but the principle's still pretty relevant, isn't it? And they wanted to know about fish populations in the lake. That's what David was particularly involved in and interested in. And actually this long-term monitoring scheme grew out of that interest and we've sustained it to this day. So now I'm going to semi-seamlessly hand over to my colleague Ellie, who's going to tell you a little bit about what these data show and what the evidence looks like at the moment, and then you'll get me again towards the end.
We have to practice with a mic, have we?
Yeah, it'll be fine.
It'll be fine. Thanks, Steve. Can everyone hear me all right? Good. Okay, I'm just going to hold it because then it's going to be easier. So as Steve said, we've got this really interesting long-term monitoring data that we've been collecting for many decades on the lake, and that allows us to try and look at some of the underlying changes that are happening, long-term changes that we can detect, and thinking about that kind of physical, chemical and biological aspects of the lake ecology, we're now going to have a few examples of some of those changes that we're seeing. So one thing in particular that we can really start to see within some of our lake time series is the effect of climate change impacting the lake. So when we think about climate change, that's not just about temperatures warming up, but also around variability in that climate signal. So when we're thinking about variability, we're talking about really intensity of storm events, so heavy rainfall, really strong winds, or also around drought, so around prolonged periods of hot weather or very low rainfall. So when we look at like an annual average trend of the surface water temperatures across those 4 lakes that Steve mentioned, what we can see is a really coherent signal of increasing water temperatures over time. So the trend of that signal is around 0.2 of a degree per decade, which is actually quite similar to an awful lot of lakes that we also know from the rest of the UK and more widely. So it's quite a consistent warming signal that's seen. And what that means is that there's been around 1 degree increase in that surface temperature of the lake between the 1950s and the 2010s. And that's the sort of really strong climate signal that we're starting to see in our data. So looking at that same surface temperature data in a slightly different way, obviously we know surface temperatures vary across the year and have this kind of seasonal pattern. And what we've done here is take a 30-year average of that surface temperature data from 1981 to 2010. So that's the dark blue lines and dots on the map, or sorry, on the graphs for each of the lakes, and a kind of measure of variability between years around that annual average, which is the kind of blue shaded area. And then superimposed over the top of that kind of long-term signal of the seasonal temperature change, we've then got a recent 5-year period of data from 2017 to 2021, which are the orange lines and dots over the top of there. And what we can see is that it's showing us where that warming is occurring. So actually what we can see there is that those orange lines and dots are slightly above that long-term trend, that long-term average more frequently in the spring and the early summer. So that's where we're seeing the warming occurring most strongly. But when we think about warming, it's not just about the surface temperatures that are affecting how the physical structure of the lake is being affected. So this is a temperature plot, but this time showing you the depth of the lake. So this is taken from one of our monitoring buoys on Blullum Tarn as an example. And what we've got here is the seasonal change in temperatures with depth. And this is to illustrate the phenomenon of thermal stratification. So this is what happens that during the winter period we have pretty much uniform water temperature, and this is the same across all 4 lake basins that we measure here, and it's a characteristic of the types of lakes that they form this stratified system. So what we get during the winter and early spring is that those temperatures are pretty much the same from the surface to the bottom of the lake. But as the warming of the sun, as it starts to warm in the spring and then into the summer, hits the surface of the water, most of that heat is absorbed there in those surface layers, and that starts to change the temperature of the surface layers, warm them up relative to the bottom of the lake, setting up this temperature difference between the top and the bottom. And because warmer temperatures means that water is more buoyant, that also then creates a density difference, which affects how things mix within the lake. And that affects things like the light climate for the algal growth, and it also affects how things are mixed within the lake, so how oxygen can move down within the water column and other chemicals and things are moved up from the sediments. So it can be a really important characteristic of trying to understand what's going on in a lake system to know what's happening with the stratification. So because we have this long-term record of temperature change in these systems, we can look at indicators of how stratification is also changing over time. And ways of doing that are by looking at things like the date at which the stratification starts in the year. So that's when the onset of stratification occurs, and also when that lake mixes then in the autumn time. So as the lakes cool because there's less heat going into them and there's more storms and wind removing heat from the lake, we eventually in the autumn time they overturn and they mix. So by looking at changes in those things, that can tell us something around how this stratification has been evolving over time. So when we're thinking about pollution, this can be from a whole range of different sources, be that from discharges, sewage works, agricultural sources, industry, or from other things like plastic and litter. But actually what we're really concentrating on here is the long-term nutrient data that we have on the lake. And that's because these nutrients, particularly nitrogen and phosphorus, are really important for driving algal growth, changing the biomass and changing the community. So starting off with phosphorus, which is the one that's most frequently kind of mentioned in relation to algal growth, what we have here are a couple of different fractions of phosphorus. So the plot on the left-hand side is a measure of total phosphorus. That's all the different forms of phosphorus in the water. And the different lines represent the different lake basins that we monitor. And then the plot on the right-hand side is what we call soluble reactive phosphorus, but it's also referred to as orthophosphate and phosphate, or dissolved phosphorus, by different organisations, and that has a slight methodological difference in how these things are measured, but essentially they're all different ways of measuring phosphorus dissolved within the water. And the reason we're interested in that fraction particularly is because it's thought to be the one that's most available to the algae, and it really boosts algal growth. They can access it very readily. So what we see in terms of long-term changes in our phosphorus concentrations in the lake over time is that typically those concentrations peaked back in the 1990s and have declined since that period. But also you notice those lines really jump around quite a lot, and that's because we get a lot of interannual variability in these data, which relates a lot to the types of the weather conditions they're experiencing, and also the different sources of these nutrients when they come into the water. So another nutrient that's also very important for the growth of algae is nitrogen. And again, within the time series, we have a long-term record of two different forms of nitrogen. Now, these are both dissolved forms of nitrogen, again, because originally they were thought to be the ones that were most important for predicting algal growth. So on the left-hand side, we've got a measure of nitrate nitrogen, and on the right-hand side, we've got a measure of ammonium nitrogen. And again, what we see is that we have a similar long-term pattern. So typically the values were higher in the 1990s and have declined since then. But again, we get an awful lot of interannual variability. Interestingly, in terms of the ammonia, though, in Esthwaite and Blullum, we've seen a more recent increase in that data. And it's something that we don't fully understand why we're seeing that at the moment. It's something we need to look into in more detail. So another kind of important chemical measure in lake ecosystems is the dissolved oxygen concentration, and that's because a lot of organisms in the lake are really dependent on good oxygen concentrations to persist and to thrive. And particularly some of the more sensitive fish species like the Arctic charr really require quite high oxygen concentrations at particular life stages. And so, and during the year, because of the stratification that occurs in the lakes, very often in the deep water, oxygen can be reduced because the deeper water is separated from the atmosphere, so there's no new oxygen getting down there, And because organic matter in the lake consumes oxygen as it's decomposed, that then reduces how much oxygen is present in deeper depths. So one really useful indicator of the oxygen availability in a lake ecosystem is to look at the annual minimum oxygen concentration that we find in those deep waters. And what we see when we look at across our 4 lake basins is that actually we have data with full oxygen profiles from about the late 1960s onwards, and actually Esthwaite and Bullen over that time have always had these really low oxygen concentrations in those deep waters. So they've been impacted for a very long time and had very low oxygen concentrations. When we look at the 2 Windermere basins, which are the 2 plots we've got here on the right-hand side, we can see a significant decline in that minimum oxygen concentration over time in both basins. So moving on then to look a bit more at the biology of the lake, and talk briefly around sort of algal blooms, which is obviously a really important topic. What I'd like to say is that not all algal blooms are bad, and actually algae form the base of the food web in lake ecosystems, so they're really, really important. So the picture of the algae in the top left-hand corner is a photographed down a microscope of a really important group of algae which tend to bloom during the springtime. So these are diatoms. And they form a really important food source for a lot of the small microscopic animals like zooplankton, which then feed upon them, which are then in turn fed upon by different fish species, and then that goes up the food web up to the large vertebrates that we know well, like the otter and the osprey, the birds and those kind of things. So they're really, really important for kick-starting the kind of seasonal pattern of growth and life in the lake. But of course, algal blooms are, we know that they can be a problem. And the photograph in the bottom left-hand corner is a picture of some cyanobacteria, so these blue-green algae, which form the kind of green scums that we can see very often along the lake shorelines. So the photographs there on the bottom and the map on the right-hand side are all taken from the Blooming Algae app. So they're records that we've had submitted by citizen scientists over the year that we've seen, and they occur, and the reason that these algae are a problem is that they can produce toxins, and they tend to accumulate in areas where people tend to go because they have this ability to be buoyant and be blown by the wind. Another way of looking at algae is to look at how much there is in the water and whether that's changed over time. So these are sort of similar plots to what I showed you with the seasonal pattern in temperature. Algae have a really distinct seasonal pattern of growth. And what we have here is, again, that 30-year average period from 1981 to 2010, which are the dark green lines and dots. And then around that green shaded area is that kind of interannual variability across that 30-year period. And then imposed over that in the slightly lighter green colour is the recent 2017 to 2021 data, which shows you how that compares over time. And what we see is there's not been a huge amount of change in those in the amount of algae that we're detecting growing in the lake over that time period. Maybe we see a little bit of a reduction in the algal concentrations in Blull and Tarn and in the south basin of Windermere, and a slight increase then in Esthwaite water and in north basin of Windermere. But these things are really variable, and it really depends on the weather conditions and the specific conditions in each year to determine how well they grow. And so you get this really big interannual variability that we see, but there is no really strong long-term trend. The interesting thing around the 2 basins of Windermere, so we were talking about how they're kind of distinct, that actually what we've seen in terms of the algal biomass is actually those have started to converge. So they're much more similar now than perhaps they were in the past.
So I'm going to take a couple of jumps up now through the aquatic food web, several jumps actually, to think about fish populations in Windermere. So you've seen them mentioned already, but I'm sure you'd already heard of Arctic charr, really an iconic cold water fish species that's present in Windermere. Arctic charr are at the very lower limit of their latitudinal range in this part of the world. Actually, they're much more characteristic of colder regions much further north. Now, what we can see of the Arctic charr in Windermere, we have catch data for the fish in both basins of the lake. And I think it's pretty clear that you can see charr catches have gone down over time and quite dramatically. Now, CPUE, if you've not come across that before, is catch per unit effort. All that is that you have to standardise the number of fish that are caught by the amount of effort that's put into fishing for them. So the number of rods, the number of nets, the amount of time spent fishing. Only by doing that can you actually compare catches from one year to the next. So that done, you can see a pretty clear decline in the charr populations. Now, of course, this fish has a great cultural significance here as well, and it's the focus of a traditional fishery as well, with plumb lines, the sort of boat-based fishery that I'm sure several of you are aware of. Now, there are several factors that are probably at play here and causing this change to happen, which really relate to what Ellie was telling you about. So we've seen that the waters are warming up. Arctic charr as a cold water fish species are very sensitive to warmer water temperatures, especially in their egg and larval stages. That's when they're particularly sensitive. At the same time, they have quite high requirements for dissolved oxygen concentrations. We've just seen that oxygen concentrations are declining in the lake as well. And then other things are happening too. So we heard about non-native species from Lindsay right at the start. So one of those, the ruff, which is a sort of distant relative of the perch, is actually an egg predator. It feeds on the eggs of other fish species. And that could be happening too. We don't really know. But the ruff are in the lake, and that could potentially be happening. So there are several things that are happening here that are potentially impacting these fish populations. And actually what we really need to do is to do some more recent surveys to try and find out what the situation is now, but crucially try and tease apart all those different effects and factors to understand what it is that's causing these changes primarily, or if it's just sort of death by a thousand cuts, really. Okay, so now I just want to think a bit more ecosystem scale about things and about the connectedness of the things that we talked about. So I guess several of you might be aware of the science of phenology, signs of spring, the kind of thing that Spring Watch introduces us to and talks us through year on year. So it's that study of when in the year swallows arrive back in the UK, when your favourite flower goes into bloom, when the oak trees bud burst, all of that sort of stuff. As you've seen, lakes are really seasonal too. Seasons happen underwater as well. And actually, if you look at the timing of seasonal events underwater, you're seeing similar shifts to the kinds of things that we're seeing on dry land. So the top plot up there on the far left-hand side, it's just several different measures of the timing of the time of year at which spring algal blooms, spring diatom blooms happen. So we've got a picture of one of the diatoms there, and I'm going to say it, I think they're rather beautiful. That's Asterianella. Now, you can see that actually, using lots of different measures of when those blooms occur, those blooms have been getting earlier over time. On average, with interannual variation, 1 year from the next, there are differences, of course. Then we come down the slide. The water fleas, or the crustacean zooplankton that feed upon the algae, so that's a Daphnia in the middle there, their seasonal peaks, which happen in the springtime, are also shifting earlier over the long term. So we're seeing a similar situation there as well. And then we come down again to the perch, which are actually numerically the most dominant fish in Windermere. Spawning times for perch have shifted earlier too, over the same period of time. Now, this is due to a complex mixture of factors. Climate is part of this as well as other factors. But the crucial point is that not all of these seasonal changes are happening at the same rate. They're changing their timing at different rates. And that can then put pressure and stress on seasonal interactions between species that are really important. So if you think about the perch, if you think about perch larvae when they initially hatch from the egg, they use up their yolk reserves and they start to feed. They need a really abundant supply of plankton to feed on so they can grow and they can survive. Now, if you have these changes in seasonality and they're different for different organisms, it could be that perch hatch too early or too late compared to seasonal peaks in their food resources. That means they don't get enough food to grow and to survive, and you can get mortality and poorer growth in perch populations because of that. And we've actually done some modelling, really, of these long-term data, and we can see a discernible effect of that on perch growth in Windermere, amongst the many other things that are affecting the population. So this is one of those other things, sort of complex ecosystem changes that aren't really very apparent from looking at the lake from the shoreline, but they absolutely happen here, just like on dry land. So I'm going to bring this talk to a close then. So what we really wanted to do is to put some evidence in front of you to show you what we understand about change in the lake and what the potential causes of those changes might be. And a really strong message is that Windermere is changing, yes, but it always has been, and over several different scales of time. There's a huge amount of evidence that we can use to show patterns of change and to try and pick apart what the drivers of those changes might be. And crucially, we can actually see impacts of climate change on Windermere already with the data we already have. You remember those changes in temperature and the timing of stratification. Water chemistry has changed, yes. We've seen decline in oxygen concentrations as well over this long period of time. We've seen changing nutrient concentrations that have kind of risen and fallen once again over the period we've been monitoring. We've seen that algal populations do form peaks and blooms at certain times of year and always have done, but there's an awful lot of variation from one year to the next in the size of those blooms and when they occur. There's a big story here about Arctic charr populations in the lake and how they may be faring. And you've seen, hopefully, that actually there is this other thing that maybe you've not thought about before, where the underwater seasons are shifting as well as a result of all of these different things that are acting on the lake environment. I think we've got one of those QR slides as well. So if you want to download the Blooming Algae app, where you can go record algal scums along the shoreline and build up the evidence base for where and when they occur, you can do that by the QR code at the top. And you can also access some of our blogs written about the state of the lake, which again shows some of these data, so you can sort of take that away and have a bit more of a read and a digest of that afterwards as well if you want to.